daar werkt de opname. All right, I think I managed to record this session, so let's go. Welcome everyone to the final end of the year Town Hall of Arise. We're going to look back at an amazing year. And of course, like the last Town Halls, we're going to start first and foremost with some user perspectives. So before we do that, just a quick view on why we're actually doing what we're doing. Arise is now alive and kicking for about 4 years. And our main objective is, of course, can we have all the information around all species in the Netherlands so that we can also recognize them in the field based on DNA, sensor technology, environmental DNA as well. And of course, that all comes together using not just DNA itself or sensors itself, but also all the data management, AI, workflows, processes, and most and foremost, people to actually get this done. So core component as well is, of course, building that reference database so that we can do the identification in the field. And that has been our main objectives, to really get the workflows going for creating such a species reference database. And now we are at a phase, and you'll see after this presentation how far we got. And I think we are now really ramping up, getting users there, and getting ready for big action. So just a quick reminder of those 2 flows. You'll see them back when we do the team updates. But first, I'd love to hear more about an actual user, an actual researcher that has been using both of these flows in her own research. So without too much ado, Emily, I hope you're ready.
Yep, I am. Hi, everyone. So yeah, I'm Emily Burdfield-Steel. I'm an end user. So I'm going to start by just quickly going through a little bit of background on my actual research question, and then talk about how Arise is helping me to answer that. So how do I move these slides? Or do I have to ask Jack? There you go. So the big focus of my research is aposematism, or warning displays. So these are commonly bright colors. They actually don't have to be bright colors, but that's also what I'm studying in this case, so we'll stick with that. And they are signals that species use to communicate to would-be predators that they are unprofitable in some way. So it's a combination of some kind of signal with some kind of secondary defense. Often chemical defense is what we're talking about here. And crucially, an association forms in the mind of the predator that when they see this signal, it's not worth attacking this prey. Next. Thank you. And I'm very interested in how the urban environment might affect these warning signals. So why, you might ask, do we expect that the urban environment is going to affect these warning signals? Well, one key component of aposematic signaling is that aposematic prey are expected to be under positive frequency dependent selection, because in order for predators to avoid these signals, they need to be familiar with them. And in many predator cases, that actually means they have to learn them. Sometimes there's innate avoidance of certain colors, but often this still needs to be backed up with learning. And the more you interact with, the more you encounter the defended prey or unprofitable prey carrying this signal, the more you will learn. So the more common you are, the better you are protected. And of course, if your population starts to go down for any reason, that can influence your protection if you're aposematic. And the second one is that in order to work, the warning signals really, they only exist in the mind of the predator. It's this association between the signal and the defense that makes them effective. Otherwise, they're just a color. Why would anyone avoid that? And we know that predators in their decision-making can be affected by many things. Next slide, please. Yep. Oh, yep, that's just a bird appearing. And for example, we know, and here I'm talking about birds a lot, because for visually signaling aposematic prey, birds are the big predators. They're the main ones because they're visual hunters. And we know that they make trade-offs, for example, when attacking prey. So it's not just the experience they've had in their life that affects whether they'll attack a defended prey. It's also how they themselves are doing. And we know that, for example, they will trade off certain amounts of chemical defense against nutritional content. So if a prey is a bit toxic but very nutritious, they may still attack it. And their body condition will also affect how they make these decisions. All of which are things that could be affected by urbanization, when birds often suffer from a lack of food. So all of this together means if there's any event of any kind reducing the aposematic population in an area, this could then lead to reduced predator avoidance learning, leading to higher predation pressure, further reducing the population, further reducing the avoidance learning, and so they can end up in this kind of death spiral. And we've seen this from literature in disturbed rainforests, for example, that the aposematic species of butterfly are often the first to disappear. So we think this could be happening in the urban environment, and we started doing some predation experiments. These are little model caterpillars. And indeed, as we expected, as you went from more rural to more urban areas in the Netherlands, the attack rate on all of these caterpillars went up, but it went up the steepest, the most on the aposematic ones. And we know from this kind of research as well that it doesn't seem to be changes in the bird community driving this. So it's similar bird communities, but rather something is changing in the bird's behavior. So we're interested to know whether they are actually getting less picky about the food they eat in urban areas and attacking more aposematic prey. And that leads me on to where Arise comes in. So this is a top-down view of Utrecht, and each of these dots is a nest box that we have in Utrecht. And the color of the dots indicates how urban the environment they are in. So the very red ones are around the central train station, very urban. So we've been monitoring this series of nest boxes in collaboration with the VU and the NEO for a couple of years. If we go to the next slide. And we know that, unsurprisingly, birds are suffering in the urban environment. So this is the survival rate of chicks. As urbanization increases, chick survival nosedives. This is consistent across years. And this is just an example, a more rural box with 14 chicks in it versus a more urban box near Utrecht Central Station, which had just 4 and fledged only 3 chicks that year. And so we think this is due to the quality of food, but we wanted to really look at what are these birds actually eating to test 2 things. Are they just getting less picky due to lack of food? And are they then therefore bringing more aposematic prey into the nest boxes? So to look at this is traditionally you just film the boxes and then you get a student to sit through the film. But I hate watching videos and I don't have the storage to store days and days and days and days worth of footage. So instead, we've been working with Yakko to use the new Ecomoni nest box cameras. So if we go to the next slide. Oh, it's gone white for me. So these sit on top of the box, and then when the bird enters the box, it takes a short video of the bird entering, and it's focused then, the camera's focused on what prey the bird is actually bringing in. And in this case, this is an example from another nest box next to Utrecht Central Station with only 1 chick. This chick did fledge though, so that was nice. And here you can see they're bringing in a chafer beetle and feeding it to the chick. Also fun in this, you can see that the adult has a tick attached to its face, so you could also even look at parasitism this way. Extratropic interactions. And through then, we've been training. You'll hear a lot more about this soon, but me and my students have been helping to then train an algorithm to automatically classify the prey that's being brought into these boxes. So far, it's very good at detecting caterpillar versus non-caterpillar, and we're working to improve this even further to finer categories. But you can already see, this was data from last spring, that the very urban boxes, several of them we had cameras on, are collecting much fewer caterpillars. So the diet of the chicks is made up less of caterpillars in the very urban areas compared to the more rural areas. And this is probably not so good. It's probably explaining the lower chick survival because caterpillars are what they should be eating, but also showing that the predation pressure on the local arthropod community is going to shift as the birds are moving away from their traditional diet and attacking more different kinds of prey. And then one more slide. Yeah, we've also been trying to combine this with DNA metabarcoding. So when we're monitoring the chicks, we also take them for weighing at certain points in their development. And here, when they poop, we can collect their feces and extract DNA from this. And from this, we can get a snapshot of what the birds have been eating that day, essentially. And this is really nice because we can compare it to the data that we get from the cameras. But it has some limitations because it really is just a snapshot, and we can't be taking the birds out of the nest every day to see what they're eating. So the nest cameras allow us to get a much more continuous long-term view on the diet, whereas this gives us a single snapshot. And if we click through the animations, I think you can see, yeah, everything's moving around. We already see from this, we have a lot of winter moths in the diet, which is represent these green caterpillars. We also saw a lot of crane flies, which by comparing then with the footage from the cameras, we could tell was actually from the crane fly larvae, not the adult crane flies. And we've also started identifying some aposematic bug species, including Ligea simulans, in the diet of the chicks. So our hope is that by combining, here you can see the snapshot. Yep, we go back to that graph. Here you can see this is just the nest boxes that we had the DNA diet from last year. And you can see in this snapshot, we get a little bit of an idea of these diptera increasing in the urban environment, but we don't get anything like as much data out of this as we do out of the cameras. So this is not so useful for really looking at the diet over time, but rather it's a nice comparison to have with the camera data. So we hope that by putting these two sources of data together, that we can get a really comprehensive view of how the diet and therefore the food web is shifting in these urban environments and what the consequences could be both for the birds and, in my interest, for the insect communities that they're preying on. That's my little spiel. Yeah.
Thanks so much, Emily. Fantastic. Very nice and concise, and for me, comprehensible, and super cool to see this happening. Thanks so much for your contribution. Oh, there's more animations.
I'm just going to share my screen quickly, Elaine. I think that's easier.
If you wish, yes.
Yeah. Okay, yeah, so I think this is a nice hook to talk a little bit about Ecomoni. So I think most of you know me as the team lead of DSI. So I'll talk about that in a minute as well. But now I would like to introduce you to the Ecomoni and how we are trying to revolutionize biodiversity monitoring a bit more broadly. So I'll first start to talk about some challenges. So basically this started with Emily contacting Twente for can we make this bird monitoring easier? And together with the UvA MDS team, we gained a lot of experience in biodiversity monitoring using all sorts of sensors. The MDS team will present this in a minute. So what I'm going to talk about is really the first step, using all sorts of sensors in the field. And you can imagine if you use a lot of them, over hundreds, it might get frustrating very quickly if the sensors don't cooperate, and often they don't. There's still many challenges in biodiversity monitoring. So first of all, there's a limited range of species. So what Emily just presented is very novel. I think a lot of species that the birds bring in haven't been seen before. So this is really a new sensors with a new range of species that can be detected. And I think there are many more to do. Many existing systems, especially when we started this 2 or 3 years ago, are still unreliable or very costly or very tied to specific vendors. For example, they don't integrate with Arise yet. There's a lot of operational constraints. It can be very time consuming and challenging to deploy them at scale. They often need frequent maintenance. So for example, due to lack of power or network coverage, this is very expensive in terms of time, but also limits the scaling of it, especially for long-term monitoring. Also inefficient data handling, waste energy and resources. So for example, if you think about managing hundreds of devices in the field, there's often no power outlets in the forest or in natural areas. So you have to deal with batteries or solar power, but solar power doesn't always work in all areas. Data integrity is a big issue. So for example, when you look at the data, was there really no activity, or was the sensor actually turned off or malfunctioning? Right? It's really important to know the difference. Same for data quality. If you deploy a sensor and leave it there for without human oversight, you're not really sure always if the camera view was okay, or there could be light issues or sound issues. Something can start growing in front of your camera. It can be condensation, or the sensor may fall over. And there's all sorts of reasons why your data quality may be compromised. So I'd like to think of biodiversity monitoring like this, right? Pervasive sensors, not too many, and an ecologist drinking a cocktail while looking at a dashboard and collecting biodiversity data. So this is basically how we started with the Ecomoni. There's a lot of opportunities that are enabled by intelligent sensors. So imagine sensors that all you have to do is plug in a battery or a power source and it starts monitoring without any hassle. Remote management allows you to configure devices remotely and also add updates to your devices. This really helps in maintaining your deployed sensor network. Proactive sensor help really helps monitoring your sensors or your sensor network over time. And self-healing capabilities allow your sensors to recover if something goes wrong without any human intervention. Things like context-aware sampling allow your sensors to adjust sampling rates based on real-time events in the environment. So in nest monitoring, for example, you could enable the sensor when a nest starts becoming active. So when the eggs hatch, or only record when something interesting happens. So in Emily, her case, that is when a bird enters with prey, that's when you want to capture a video and otherwise not. This really saves a lot of data and time. Most of all, intelligent sensors provide actionable insights, right? When something happens, you may want to be alerted so you can take action. So this is why we started Ecomoni, I think 2 years ago. This came out of the Ecomoni, and the idea is to really scale biodiversity monitoring and develop hardware and a platform around it to enable large-scale, high-throughput monitoring. So I'll go give a brief insight into the Ecomoni and where we are now, and this will nicely tie together with the Arise update. So the Ecomoni, the goal is to automatically count and classify prey. That basically you hang it up, and at the end you should get a report of the prey that was brought in by the birds. At the same time, we've developed a platform that really allows you to configure and manage hundreds of devices. You can make groups of devices and configure them individually or per group. The Ecomonies, they integrate with Arise. So what you see here is a snapshot of the MDS portal developed at UvA. So the videos from the Ecomonies are directly pushed to this portal. And there's also a new client that can push the videos directly to the BioCloud, which will develop further next year. This is the kind of videos that are collected. I think Emily already showed a few snapshots. There's a large variety of nest boxes, backgrounds, and a variety of prey. So just to give you an idea of what we've seen so far. During the nesting period, we can see different types of nest material that's brought in. There's some researchers interested in that. Obviously, a lot of caterpillars. Then a lot of spiders, and different types of spiders, but also spider eggs. And yeah, some of these, they actually collect the spider and the spider eggs together. It's a nice combo meal. We see a lot of beetle larvae, but also yeah, weird things like ticks. A lot of pupas and different colors and varieties. And yeah, just some random snapshots of unknown stuff, but crickets, butterflies, moths, you name it. And of course, what comes in must go out. So it's actually quite doable to detect feces, which Emily mentioned could also be a good proxy for the amount of food that they have available. So a little insight into how the Ecomoni dashboard looks. So the system allows you to configure groups of devices and yeah, basically change everything you need to change, like light settings, camera settings, upload settings, and time schedules. So the devices can be turned off to lengthen your deployment period to save battery. Basically, they can, for example, they can start with sunrise or sunset, you name it. So I think this is really important. The devices, they record and send health updates. This will tell you that the device was actually turned on and actually monitoring. And yeah, in these packets, we can add all the information we want. For example, the storage or the temperature, storage space or the temperature of the device. And packages like this we can also use to send updates like an entering event or other events of interest. They are displayed on the dashboard in real time. Yeah, this is basically showing you that information that I just mentioned. So the location of the device, the software version is running, all nicely presented on the dashboard. Now, dashboards are nice, and if you collect a lot of data, this gets overwhelming really quickly and frustrating again, and that's where AI comes in. So also within Ecomoni, we are developing several algorithms. So it's not just one algorithm, but it's actually a collection of algorithms. So for example, first, seems basic, but it's quite interesting to us to detect the entrance, because this we can use to know where we need to look and already downsize the data we need to analyze. So if you look only at the entrance location of the video, this allows us to run very small models to, for example, do entry detection. So why do you want entry detection? Well, here you see a video of a bird exiting, and it's very cute, but yeah, nothing really interesting happens here. So these kind of videos are triggered by the sensor on the outside, but we also want to filter them. And that's why we have an algorithm that detects entry. So we can already make an additional filtering on the videos. Another model we're training is insect detection. So this allows us to trigger only when there's an insect in view. That is even more specific. So when you really want to look at only insects, then you can even filter out entry videos for insect videos, and so on. So we have a caterpillar classifier and a multi-prey classifier. In the end, we can make graphs like this. So here you see data over time. The green shaded areas are the periods where the device was actually monitoring, where we have the health packets. We know for sure it was recording. So for example, here on the 6th of May, in the middle of the day, it stopped recording. So you could think, oh, maybe the nest fledged. But actually in that case, something was wrong with the device and there was no health packets. So we know it was compromised. But here on the 13th of May, you see something similar, but there the nest actually fledged. So after that day, the nest box was empty. So the blue bars here are the actual number of recordings. Then the green bars are the number of entries, so it's a little bit less. And then orange is the number of detected caterpillars, and purple is other prey. And these this distinction of the actual number of caterpillars and other, they will get better and better as the algorithms improve. Of course, you can also aggregate this over a period to make nice pie charts. And we are working on the insect classifier to get more and more fine-grained classification of the prey being brought in. So far, they have been used in the Amsterdam Flevo Park in Utrecht by Emily, but the MDS team has also deployed them at the Arctic Zoo in Amsterdam and at Hoge Veluwe in collaboration with the NEO. And we have deployed them with the county of Hengelo, and of course here in my backyard in Pijs as well. So getting closer to the ARISE update again. This is the training data we have annotated ourselves so far together with Emily and her students. As you can see, most of the training data are caterpillars, and then a long tail of other stuff. There's only 8 ticks so far, because this is a really expensive, a really tedious part, getting training data, and it really affects the performance of the algorithm. And as many other data sets, it has a long tail distribution, which is always a challenge. So what we do right now is we use our caterpillar classifier to already filter out the caterpillars. So when we annotate data, we really focus on the rare classes. So the stuff we annotate right now is already quite interesting because we don't have to go through all those caterpillars, really speeding up this work. And this is a collaborative effort, right? Would be nice if we could always do it in an environment like this. But I think what Emily and myself so far is showing that this is actually something we can do together. And when we collaborate across teams, so everyone basically wants to use Ecomoni, can also help in improving the training data. And this is where I'll come back to later. So this was the Ecomoni update so far. And I will now hand over to Emil.
Thank you. Yeah, I will briefly go over some of the highlights from the field where the sensors were deployed. And here we see an example of one of those that only looks, the diopsis that only looks at insects, with the yellow screen where we can see some of the insects, which are then later on identified in a similar vein as Jacob already explained for the nest monies. And while there are still many challenges, thank you. Yeah, there is one of those also mentioned by Jacob is the fact that there are so many sensors that we have. So we observe the different groups of insects, flying insects with different types of equipment to also see how they capture different parts of the biodiversity. We see at the left-hand side, apart from the diopsis, also the hoverfly detection, the moth detection cameras and classification devices, as well as specific pollinators on sedum vegetations. And then apart from the nest monies, we have also, of course, the wildlife cameras, as well as one on the connected to the radar. So when the bird scan radar observes signals of wingbeat frequency, we can match those with video footage to identify the birds, and acoustic sensors in different spectra, so to identify birds and bats. And all of these in different amounts in the field where some of the data is downloaded in batches and classified and analyzed offline, but eventually the aim is to push all of these through the BioCloud as well, and to find a more organized and homogeneous way to process this. One of the highlights from the field was collected mainly in the two sites, the Amsterdamse Waterleidingduinen and the Oostvaardersplassen, where most of these devices are deployed. At the upper right, you see a time series of geese that were detected by wildlife cameras, and so not only detected but also counted, with the ultimate aim to not only count them but also assess grazing density in relation to vegetation surveys. So that's to turn the biodiversity observation of birds into other ecological relevant data. So, and that's a process that works well, that has been so far been very successful and automated successfully. And at the lower left there, we have matching between traditional surveys of fallow deer counts in the Amsterdamse Waterleidingduinen and the counts as derived from the network of wildlife cameras. It's not trivial to have similar outcomes for the two different methods, but here it turned out to work really well, thanks to, I think, a very thoughtful design of where the cameras are placed and a sufficient amount of cameras. You see here different boxes refer to different vegetation, so the different environments also match in density of observed fallow deer. We have some plans for the future. Let's go on to the next slide. The aim for the coming year is to stay doing this, what we're doing in these sites, but try to connect to other monitoring programs that are ongoing. Amongst others, there is one by the Dutch Environmental Assessment Agency, part of RIVM. They have a monitoring program called IMN. It is measuring mostly environmental factors. Here we see the famous nitrogen loads in the environment. And we would like to connect our measurements to that network, because there should be a relationship, of course, between biodiversity and these pressures, as well as the National Ecologically Monitoring Network, called NEM, coordinated by CBS, and the activities that are ongoing in the long-term ecological monitoring site. One of these is the Veluwe, and we also deploy some of our equipment there. We also have plans, amongst others, with acoustic devices and playing around with some newer developments. And one of those, if we get a chance, I'm not sure, we'll definitely also look into possibilities for autonomous monitoring. It's not sure that we will put a robot like this one in action, but we will definitely try to team up with a team that has this kind of equipment and see how we could make use of that. We have to think about vegetation mostly in that case. This was my last slide, I think, so we can move on to the next group.
Yeah, that's me again. Thanks, Emil.
Yeah.
It's, the robot looks very nice. It's exciting.
Well, it's an industrial. This one, industrial produced and for industrial monitoring, and also being put to work in biodiversity settings.
Awesome. Yeah, looking forward to hear more about that. Yes, I'll give a brief highlight of the DSI team, the Digital Species Identification team. So this year, I think a lot happened. So I pulled out the confetti slide again. This was to celebrate that we actually have a public URL, which for a long time we didn't have. So if you go to this URL, you can actually sign up to join the DSI app. Of course, you have to be added as a user first. We're doing this staged, but the fact we can is also another milestone. So we have access management, and we can now organize who can see what. So another big update is that you can now actually organize your stuff in projects, and these projects are synchronized with Faunabit that produces the insect cameras, and also with the MDS portal. So all the projects that have been made in the MDS portal are also shown here on the top right. Yeah, and a lot happened this year in the machine. So I started listing them here, but this bullet list will become very long and I think boring to most of you. But just a few highlights that the team has worked very hard on is optimizing our servers and the server loads, so that when we have more and more users, that the system actually doesn't fall over. And this is it's crunching along nicely. Also includes stuff like auto recovery of our servers. We have refined our result validation, so are the results that come out really correct, and can we detect when something goes wrong? Because if you're analyzing hundreds of thousands of media items, errors can creep in and you may not notice. So this is we bumped our heads a couple of times, but this actually made the system better this year. Also, the exporting of results is more robust, and every week, I think, as a user, you also get more insight into what's going on and more feedback to the user. So it becomes nicer to use the system. And the results, for example, are now versioned. So you can also see what type of code was used to generate the results that you have, because this also has an effect. So this list of the list of our Ecodem repository is growing. It's I think next year it won't fit on one slide anymore. It will become too long to talk about, but I think that's good news. So I should highlight the new ones actually, but the most recent ones are Trapper AI and DeepFawn that we can use to analyze wildlife camera images. I think they're practically done, not in production yet, but I think they will be soon. Aves Echo and Capgemini are the two audio classifiers that are available now to analyze audio. And the MDS team has started pushing audio data as well from AudioMOS, so we can actually, I think early next year, show an end-to-end demonstration of audio monitoring. And another notable algorithm is a Hugging Face connector. It's basically one algorithm in our system, but it will allow you to select any Hugging Face model that is available on the Hugging Face platform. Not all of you may know Hugging Face, but it is a very popular platform that hosts more than 900,000 models. Not all of them for biodiversity, of course, but it's a really interesting connection. Now, coming back to this collaboration and annotation, it doesn't just apply to Nesmoni, but it's actually a generic thing, right? So there's a lot of algorithms, but they're never perfect. And they always have a limited set of categories that they can detect well. So there's often a need to collect more training data. I'm convinced that here lies a really big opportunity for Arise. So also the use of the OBS cameras are interested in this idea. So we are now working on an integration between Arise and labeling tools. So on the right, you see a snapshot of Labelbox. It's one of many annotation tools these days. And we are working towards a seamless integration, which means you have a button on the DSI system that will create a project for you in the label box and get you annotating as quickly as possible. And vice versa. So when you're done annotating, synchronize all the annotations back to Arise so that they're nicely organized in our ecosystem. Then very quickly you arrive at this point, sharing vocabularies. So if you want to work together on data and training data, then you have to decide what labels you are using for your categories. Now this is a whole domain in itself, right? I think there's entire research projects on optimizing this, also within Naturalis. So again, that's an opportunity. And so the problem is that biodiversity data sets, they not only span hierarchical taxonomies, but also behaviors and environmental factors that occur in your projects. And if you don't organize a system around this, then you get a lot of fragmented annotations and different labels for identical categories. And this really misaligns data, but also reduces the effectiveness of algorithms. And so it lowers the quality of your AI. So there's large opportunity here to organize something around collaborative ontology management. This is where we are now. So we want this to be a modular, collaborative, version controlled, but also flexible, right? If you are working on your project and creating your training data, you still need to be in control. But at the same time, we really want to enhance collaboration on this. And I think we have some really nice ideas on how to do this. So our outlook for next year, we will start writing DSI results to BioCloud. A lot of preparation has been done for this. So I think early next year, we will actually complete the circle. Like I said, we will integrate annotation platforms, work on the collaborative ontology management, and something I have not talked about today, but it's, next year we also will start demonstrating that we can update our algorithms. Because why have training data when you're not using it to improve your AI, right? So we are already connected to Snellius, and next year we want to really start using it where it's built for, and that is to update our AI.
Cool. Thanks so much, Jacob. Will you continue harnessing the slides?
Yeah.
Awesome. Fantastic. Thanks, Ellie. So let's move a bit to the going from sample to DNA identification side of Arise. Of course, a lot of things happen there as well. And first and foremost, all the cool stuff that has been happening in the sampling team. Yeah, there you go, Charda. Jacob, that was your cue. Thank you. Okay, of course, we've been showcasing this side of Arise a lot over the last year, but just to highlight a few of those things, the cool stuff that's there, that we really have several sample kits ready. The insect sampling kit has been handed out like over 100 times. The plant sampling kit has been used a little bit, but the mushroom sample kit definitely has been used by the Mycological Society, so that's great news. It seems like a low number. We have 20 great users on ADA, our sampling tool, meaning that 80% of our users are still handing in sheets, but we're slowly transferring those users to use ADA to really harmonize and standardize the entry of metadata, because we've also encountered quite a lot of issues with the sheets. So this really pushes our resolve to go for a simple tool to do the registration. And this is also what will be used on a larger scale when we're talking about environmental samples in the very near future. The lab has been up and running. The team has been doing awesome, over 60,000 images. And indeed, the next slide will showcase the biggest numbers of all. Like we have 102 collectors that have identified specimen for 20,000 specimens. And next to that, we've also been trapping a lot in the wild, because of course there will be lots of different species that are a bit unloved, and we also need to find those. So the team has been really working on all of those Malay straps, and boom, here comes the number. In total, over 106,000 specimens have now been subsampled by this team. And for this year alone, it's roughly 70,000 specimens. So over the past years, we've really ramped up the workflows. They've done an amazing job. And this is the only way that we can really get that species reference list together. So thanks a lot to that team. Kudos. So outlook, of course, a lot more needs to be done because even with those numbers, we see only small increases in the actual species that we can now scratch off our target list. So there will be more targeted approaches, really talking to the communities behind it, and looking for particular species groups to fill the gaps, and increasing our speed. So right now the team has already improved, and from getting a sample kit into actual processing is roughly 3 months now, and of course we're going to drop those numbers more and more. So thanks, team.
Okay. Yeah. So we're a little bit behind with the sequencing part, mainly because of all these specimens coming in this year. But we did a tally, and there's over 50,000 DNA extracts ready and done now, with about almost 90% amplification success. So we're doing pretty well. And then I also fetched some additional numbers out of all these samples. So at any stage in this processing now at Naturalis, there is more than 14,000 specimens with species level identification. And so that's actually things that are on a Dutch species list and not just Insecta spec. And there are about 4,500 different species. I have to say the majority is still animals. So we need to get some more plants and fungi into the pipeline, but I think that it's a work in progress. We've been working a lot on the capacity here at Naturalis. So we can now do 24 plates. So that's over 2,000 samples per week. So that's pretty impressive. But also Westerdijk is not sitting on their hands. And so in the last couple of months, they've been doing a lot of actual culturing of strains from soil samples, so about 100 new strains per week, what I can see from the average numbers that were reported. And that's a lot of work, like culturing the samples, sequencing several markers, characterizing all the species. So it's also pretty impressive. And then we've been working on some of the legacy data that we still have lying around, because we have a freezer with tens of thousands of DNA extracts from earlier projects for which we don't all have sequences. And so now we are working also on resequencing some of those samples, just getting all the samples from the -80 freezer. And the first tests are about 75% success. So cautiously optimistic, but at least for the plants where we started, that's potentially hundreds of new sequences. So lots of more species that we can hopefully cross off the target list soon. And then, of course, we cannot have a town hall without these wonderful plates of all the new species that are being described by Westerdijk. So here's just a selection of what's been described. I'm not going to name them all, but if you have any questions, there's people from Westerdijk in the audience, so please feel free to contact them. And then...
Yeah, so the sequencing of fresh material in the ARISE project is being done on the Oxford Nanopore platform, and out of that comes raw data, and so that needs some amount of post-processing. We've had a pipeline that does that, sort of in production for a while now. That's a command line based tool, so operated by experts, let's say, quite tightly integrated in the rest of the chain. The recent development is that we actually want to give access to this functionality via other entry points so that researchers can use this as well. For this, we use the Galaxy platform. So Galaxy is a web-based tool to do bioinformatics with just point and click, so it's quite user-friendly. And so recently, we basically to try this out and to show it to our colleagues, we did a workshop during the Research and Education Day, and that's what this photo is from. So a bunch of different researchers assembled their own barcode sequence from raw nanopore data, and that was easy for them to do because it's an easier interface. Nice thing, I mean, the platform has many advantages that I won't go into, but one of them is that there's pretty easy authentication for users. So within Naturalis, we use this Azure-based two-factor authentication system. You don't, as a Naturalis user, you don't have to register any special account or whatever. If you have your Naturalis account, you can get in. But also, the authentication is also possible via this federated system called SRAM, which goes via SURF. So any Dutch educational research institutes connected to SURF can also get on this. And so recently we did a bit of a stress test with this class that I've been teaching at the university here in Leiden. So none of those students obviously have Naturalis accounts, but they have Leiden accounts. And so they could also get on Galaxy, and 4 students at the same time ran this quite small meta barcoding pipeline. And the system didn't break a sweat, so that's nice. It's all very stable and performant. Actually, there's in the backend, there's the infra wizard who can just add extra compute nodes to it. So that's how it absorbs the extra loads. Another thing that's nice about this platform is that it has a web service interface. So that also means that elsewhere in our larger infrastructure, people can write code that basically accesses this functionality for high throughput kind of processes. And we see that as probably the way forward for the integration of larger scale infrastructure. And we have that now available for this barcode processing pipeline, but there's others in the work. So one thing that needs to happen, regardless, or basically for many sequencing platforms, not just Nanopore, is validation steps. So you've built your consensus sequence. Here, in this case, it's a consensus sequence. With genome skimming, it's obtained another way. But in any case, there's your quantitative sequence that you want to check. And broadly speaking, there's 2 different things that you look for. So there's a structural validation, where you basically look at, is my sequence long enough? Does it not have too many ambiguous codes? So beyond A, C, G, and T, there's these other letters promulgated by the International Union of Pure and Applied Chemists, which map onto greater or lesser levels of ambiguity, and you don't want a lot of that. So part of the structural check is to look for that. Another part is if it's a coding gene, so it translates into an amino acid, there must not be an early stop codon, because that would mean it's probably a pseudo gene that you sequenced instead. So those kind of checks, that all falls under structural validation, which is basically something you do once because, you know, it either passes that or fails. The other thing that we want to do, and that we are doing, is basically taxonomy checks. So is my sequence actually close to the species that we expected? And that you want to do iteratively, because this is in relation to reference databases, which gradually get finer and finer. Both of these functionalities are moving toward Galaxy as well. Now, lastly, 2 other things that are work in progress. Meta barcoding pipeline, work in progress. Recently, we applied that to the Arise Soil meta barcoding pilot library, just to pick different sequencer platforms actually for a different project, but it was nice to have a functional initial version, minimal viable product, let's say. And likewise for library curation, so we have a basic pipeline that we ran on Bold, and this is gradually being expanded, firstly to look more also at sequence quality within Bold, and secondly to also be applicable to other sources of reference data, for example, Unite. And that's what we got. Thank you.
All right, all that data. So all the data that the other teams are generating and collecting needs to be managed somehow. And for this, we have a big system with 2 different data models, as we call it. So all the DNA related data goes into the core sequence cloud, and all the sensor AI related data goes into the bio cloud. And both live in the same system. So from there, we can serve this data to consumers, which at the moment is mostly our own ARISE digital species identification portal and our DNA portal, of which I will tell a bit more in a bit. But in the future, we also would like to send that to other infrastructures. So what happened in this year? At the start of the year, we had one Diopsis data pipeline. We've been talking a lot about these Diopsis insect images, and that pipeline really set the basis for how we want to actually ingest that data, manage that data, harmonize it. At the moment, we work really hard this year more on the data pipelines for the DNA-based data. So we actually added 3 data pipelines to ingest all the sample metadata that comes in through either ADA or through the sheets. People handing in data through sheets have a bit of a habit of making up their own minds about formats of a date or numbers. You would think a number is just 1, 2, 3, but can also be a dozen or things like that. So we try to harmonize and standardize all of that, as well as the nanopore sequence data. So all that data now is ingested into the core sequence cloud and published onto the DNA portal. For the sensor pipelines, we have now just started to rework the BioCloud to also be able to ingest data from multiple sources, and the sound recorder data from MDS is in a testing phase to then send that to the digital species identification portal. And they're waiting for it because, as Jacob showed, they now have algorithms available to analyze all these different media files. So we're keen to get more and more sensor data into BioCloud and push that to the digital species ID. Some numbers then, because we like numbers. I think we host now more than 28 million Diopsis insect images, but also the barcode number is going up. When I created this slide last week, I had, 8,000 barcodes, and then it went up to 14,000 yesterday, and this morning it was 19,000. So we're crunching the data that's coming out of the nanopore pipeline through the core sequence cloud as we speak. The specimen numbers are still a bit low, but once the sheets are actually updated with proper numbers, not dozens, then that number will increase very soon as well. 2 things I think that have been really important this year that are always a little bit difficult to explain, but very essential. We've implemented Keycloak as our user management system, and this really means that for both the DNA portal as well as the DSI portal, anyone can now log in. If you have a university credential, you log in with your own credentials, and we can, in the backend, link the data that is yours to that. So most of our users at the moment are either people that go to the DSI portal or to the DNA portal, but in the future, if you have both, you'll easily be able to log into both systems. And we also have a ARISE API. That's just an interface for systems to talk to each other, but very necessary for the building blocks for this data management system. Then I think the next slide is about the DNA portal. Yay! I'm doing this on behalf of the apps team. Victor's been leading this team while Lotte is on maternity leave, couldn't be here today. So the DNA portal has been launched, and this is where our ARISE samplers who do all the work to help us fill this reference database can view their data. So this is where the data collected through ADA and the sheets, and then the nanopore data are actually finally visible. Next slide, Jacob. It's still, this is kind of what it looks at the moment. So the left screenshot is a list of all the samples that have been handed in by a particular user, and when you click on a particular sample, you can see the metadata of that. And at the moment, it doesn't show the sequence yet, but that will soon be there. If you are an ARISE sampler, you might be wondering, when can I log in and see my data? At the moment, we're one by one adding users to the DNA portal. We want to make sure that this all goes smoothly, but over the next few weeks, if you're an ARISE sampler, you will get an invitation to log into this portal. This is where you can see your own samples, and the DNA analysis is kind of running always a little bit behind samples coming in. That's how the process in the lab goes. But once the DNA analysis is complete, the DNA barcodes will show up for your samples. So yeah, that was
Awesome. So thanks so much, team. And I think you're already overwhelmed by now of all the cool stuff that has happened also in the last quarter. If that wasn't enough, we, of course, we're also busy with just outreach and community building. We've had an amazing Arise Day, and yes, there will be a new edition in 2025. And we had our first official publication. We had another cool barcoding NL event with validation workshops for our experts. A first barcoding NL magazine. Here's also the digital version to that. And we've been visiting various species organizations, various stakeholders. Basically, we're trying our best to really showcase Arise and have users really interacting with it. So if you have any other ideas of how to get there, let us know. And we'll be on the road towards you. And yeah, that's it. I'll stick around a bit for some more feedback, or if you would like to leave your comments, now's your chance.